Hi, I'm Ben Chung, and today I'm going to be going over optical tweezers at the Bell Labs Museum Showcase. Now, I have other videos where I go over the entire showcase here, you can see, so be sure to check out the overview video. But in this video, I'm going to be going over a specific thing in the showcase, which is called optical tweezers. It was designed and developed by Arthur Ashkin, and it's won a Nobel Prize. So if you follow me down here, this is inventions on basic communications, AV, information technologies, and this is just about science. So one of the things that won a Bell Labs a Nobel Prize was called Optical Tweezers by Arthur Ashkin, where he's invented optical tweezers that grab particles, atoms, and viruses in 1987. This is his when he gave when he got the Nobel Prize. This is an actual copy of his talk that he gave. So optical tweezers, in this talk, he talks about this Crookes radiometer, right? So a Crookes radiometer is interesting because basically it has veins on it, and these veins can absorb light, and some of them are white and some of them are dark. So it shows that the force of light as it hits one of these veins can cause the veins in this evacuated tube to spin. And he has this insight that basically light can apply a force to something. So he looks at the forces of a laser, and the forces of a laser and as photons can actually impart a force on something, which isn't necessarily obvious. And the more powerful a light, the more force that can be imparted. And he actually does some math to calculate the forces that are impinged on something, in particular a little like spherical. And so the incoming beam of light is going to get bent. It's going to refract around the sphere, and through that bending of light that causes a force, he can actually manipulate matter. This is actually from his notebook, a figure where an excerpt of his figure in September 8th, 1969, where he talks about this optical trap idea. So basically, if we look at a laser, the center beam of a laser, and if we have he's looking at a little glass bead, and what happens to the forces, the gradient and scattering forces of that bead in a laser. But to kind of understand his work better, let's take a look at what happens. So essentially, if we have a bead and you have a light shining through it, what's going to happen is the light is going to bend in that bead. It's going to refract and reflect in that bead. But what happens is, as the light bends, it's going to cause a force. If we just look at one of these beams, it's going to cause a force against the bending, so that this beam would push the bead down, which is going to want for every force is an equal and opposite reaction. So that's going to want to push the bead this way, and this light is going to bend this way and pushing the bead that way, and the bead's going to react this way. So in a sense, you're kind of pinching that particle, that bead, and today this optical trap idea has the ability to trap individual atoms, and it's used in a lot of atomic studies. So so we can see basically there's momentum and absorption, and those play a role in how to pinch this atom together. So he calculates that basically, if we look at the refraction or reflection of the light, of the incoming light, what are the forces in play? Of course, gravity is trying to pull something down, but the light is actually pinching. You can kind of think of it as, and that's why it's called optical tweezers, kind of pinching that matter. This was from another great video that I saw, which explains this concept really well. So basically, if we look at a laser shining down on this bead or particle, as the light bends, it's going to want to push the bead this way, but of course, for every action, there's an equal and opposite reaction that the bead's going to naturally react to go this way. And if you have two of them, that's going to, one pushes this way, one pushes that way, that's going to essentially pinch the bead or particle. So in a sense, as the refraction light pushes it down, the force is going to want to push it up. So here we can see an actual picture of a paramecium where they use the internal organelles, and in that arrow you can see that he was able to manipulate the optical tweezers, and this is from his Nobel lecture. So optical tweezers are used for all sorts of things. Single molecule studies, optical tweezers enable the manipulation of biomolecules like DNA and RNA, and proteins, revolutionizing our understanding of molecular processes such as DNA. It's used in cell manipulation, in colloidal and nanoparticle manipulation. Anywhere you want to study basically an individual atom, or particularly like in biological studies, it's found applications. So, for example, studying DNA and RNA, how these optical tweezers are able to manipulate DNA and RNA so that biologists can study that. And here's kind of a setup of how that works. So basically, there's a trapping laser, a mirror, and here we see that optical trap where you're holding a sample of something, and in this case, a biomolecule. And again, that sort of pinching effect with the lasers and the various forces acting on it. And this is looking at one of the studies where, so protein folding in particular is a very interesting area in mathematics and in biology, because RNA that from our genes create amino acids, and those amino acids fold up, automatically fold up in a particular way. Our body is essentially a nanomachine. It works at 1 × 10⁻⁹ meters, and it works by a lot of things sort of bumping into each other. And so the shape of things is very important, and so protein folding is of particular interest to biologists. So we see here the actual display here. So Arthur Ashkin invented optical tweezers that grab particles, atoms, viruses, and other living cells with their laser beam fingers. And as is always the case in Bell Labs, the breakthrough came as a byproduct of research on the fundamentals of communication, and in this case, optical communications research into non-fiber optical systems. Out of his work on advanced laser optics came the ability to use optical pressure from high-powered lasers to control microscopic particles, atoms, molecules, and living cells. It was a major breakthrough that came in 1987, and when Arthur Ashkin used optical tweezers to capture living bacteria without harming them, the optical tweezers he pioneered are now widely used to investigate the machinery of life. Okay, so thank you for watching this video. There'll be more to come. I'll probably do a series on all of the Nobel Prizes. This is one of the 10 Nobel Prizes that Bell Labs has won. Be sure to check out I currently have another video on lasers, so you can check that video out too. So thank you and have a great day.